When we're talking about DNA, and this is a very unique feature of DNA that I like to talk about a little bit, that is the denaturation of DNA. Okay, if we have a native DNA, okay, so native meaning double-stranded DNA, if you scan it using UV spectrophotometer, and you're going to see that it will have a peak absorbance at 260 nanometers. Okay, and this is the wavelength that we use to quantify DNA. Okay, so if you guys work in a lab, sometimes you have to quantify the concentration of your DNA in a given sample. What you can do is take that sample, shoot it on a little thingy called nanodrop, and you'll get a measurement of UV absorbance. And then you can calculate based on how much you absorb, how much DNA you have left in your sample. This is double-stranded DNA. So when your DNA becomes denatured, when it becomes single-stranded, essentially single-stranded, so it no longer has that double helix structure, then what you notice is that your absorbance will actually increase. You will have a larger absorbance. And this feature is actually used to allow you to determine the melting temperature of your particular DNA. Okay? And the way you can do it is where you start with some amount of DNA, and let's say we have an absorbance equals to 1, and you heat up your sample. Typically, your absorbance does not change with temperature, but what you notice is that you're heating and heating and heating, and all of a sudden you have a large increase in your absorbance, and this change in your absorbance corresponds to the difference between double-stranded DNA and single-stranded DNA. Okay, so this is called a hypochromicity. This feature allows you to determine the TM of your sample, and that could be useful depending on what kind of experiments that you guys are doing. TM changes with the amount of GCs you have in your sequence, because remember that G pairs with C using 3 hydrogen bonds, and A pairs with T with 2 hydrogen bonds. And obviously, the more hydrogen bonds you have, then the stronger it is for them to glue together. That means that the more heat you have to give it in order to separate them into single-stranded DNA. Okay, so if you want to denature your DNA, you probably want to know what's the percentage of GC. Okay, so in a lot of the research that we do, we always care about the GC percent. Okay, so when you're doing PCR, for example, you need to make sure that the GC percent is not too high, because if it's too high, you have to sometimes add a little bit of organic solvent to help the DNA denature, and then you can have a successful PCR. Okay? So that's the effect of GC percent on the melting temperature for your DNA. The higher amount of GCs you have in your sample, then obviously the larger the TM will be. Okay? And that's due to the number of hydrogen bonds. Today I want to spend a little bit of time talking about recombinant DNA technology. Really, that's not the focus of this class because we're more interested in biochemistry. But since recombinant DNA is like one of the major things that you can do out there today, so we'll touch on it a little bit. You'll learn more about that when you take molecular biology. But so here it is. All right, recombinant DNA techniques are used to study nucleic acids and their transcription and translation into proteins. Right? So really, long time ago, before you have recombinant DNAs, it's actually quite difficult to study something, okay? Because you need to get a sufficient amount of DNA before you can study it, okay? Recombinant DNA does that for you. All right, so some recombinant DNA techniques include gene cloning, that's very essential. Chemical synthesis of oligonucleotides, okay? We'll see that today because you need to have small nucleotides in order to serve as primers for doing PCR. We have DNA sequence analysis, okay? To do that, you have to have DNA sequencing, okay? We'll talk about standard sequencing today. You can have site-directed mutagenesis. Let's say you want to study a particular amino acid residue in your protein, like what does it do? You probably have to have control over the DNA that makes that protein. Okay, so if you have recombinant DNA techniques, what you can do is you can make DNA mutations of that gene, and when that gene is translated into a protein, you can have a different amino acid residue, and that's how you can study, using mutations, study the function of your protein. You can also have PCR. So PCR is important because it amplifies the DNA for you. You can have homologous recombination. This is a very popular method that people use to introduce a synthetic gene or just a foreign gene into an organism. Okay, for example, when my lab do research, this is a method that we use almost all the time. And some of the applications for recombinant DNA techniques are recombinant protein production. We see that insulin, growth hormones, vaccine or protein-based vaccines can be produced using recombinant DNA techniques, okay? And insulin in particular is one of the first successful biotechnology applications out there, okay? So insulin is a protein drug that we make to treat diabetes. We also have genetic modification of crops. For example, you engineer crops to be insect and herbicide resistant, cold and drought resistant, things like that. We talked about that last time. The stuff that I do in my lab, synthetic bio, we make biofuels, biochemicals, etc. Or we can do some studying of a specific function of a gene or a protein. Okay, so you can purify human proteins, or you can purify other proteins in E. coli, and then we can study it in a large amount. And then we have a lot of other applications out there for recombinant DNA techniques. We talked about PCR last time, okay? The power of PCR is really the ability to amplify your gene, okay? So given a genome, okay, a random genome somewhere, once you have the sequence of the genome, okay, then you can identify which region you want to amplify. In order to study a function of a gene, of a DNA segment, you need to have a lot of it. So if you were to do a PCR, essentially you start with 2' DNA always go from 5' to 3', 5' to 3', and essentially what you can do is do a thermal cycling where you first denature your DNA such that they become single-stranded, and then you add your primers. It's already in the mix. The primers will anneal to your denatured DNA at a lowered temperature. And then you turn the temperature up to a temperature that is where the polymerase does its job the best. Usually for thermostable DNA polymerases, it's going to be about like 68 to 72. And then your DNA polymerase will start to polymerize the DNA. And then you recycle this process. Then there you have it. That is the PCR. So you probably learned more about PCR back in general bio, or what you will learn it again in molecular biology. Okay, so PCR is a very useful method to generate you a lot of the DNA fragment of the specific sequence. When we're creating recombinant DNA, you know, we can do a cut and paste strategy. If you guys are interested, I can show you what people do now. Okay, so increasingly people don't do this digestion and ligation method anymore, but rather they can do it using homology. Okay, but in short, what we have is DNA can be manipulated through this digestion and ligation methods, where as long as the 2 fragments of DNA have the same cut sites that are recognizable by digestion enzyme or restriction enzymes, okay? They have the same sequence. So if you treat them with the same restriction enzyme, you'll be left with the same 4 base overhang. And when those bases anneal to each other, and then you add a ligase, you link them physically together, and that is recombinant DNA. Okay, that is actually what is meant by recombinant DNA. So what you can do is you can take vectors, typically they're plasmids, okay, and you can cut them using the same enzyme that you would cut your particular gene of interest. And then you mix them together, you use ligase, and then you ligate them into one single piece of DNA. And then you throw them into E. coli, for example, and then E. coli can make whatever is encoded by your gene for you. So it's going to make a lot of it, and then that is the recombinant protein that you can purify to study, or you make recombinant proteins to do whatever that you wanted to do. Okay? So this is essentially how you create recombinant DNA. Okay? By first you amplify a fragment from PCR, and then you cut them using the same restriction enzymes such that you create a same sequence in the overhang. Okay? You cut them, and then afterwards you paste them using a ligase, okay, DNA ligase, and then they become one single DNA molecule, and then you can use E. coli to make more of that DNA for you. So this is recombinant DNA, and this is something that is done very routinely in almost every lab that studies proteins. Okay? All right, so this is what I want to get into. In order to do PCR, you have to have primers. Okay, so some of you are doing undergraduate research already, and what you notice is that a lot of times when you go and do PCR, your professor say, oh, you use this primer and that primer. Okay, primers. Primers you buy from companies. Okay, companies can synthesize primers for you. Now the question is, how do they make primers? Okay. So they actually make these primers or short oligonucleotides using a chemical synthesis, okay? And this is how they do it. The way it works, okay, is it's actually done using a solid phase synthesis. And by solid phase, it means that first you have a nucleotide, okay, that is attached to a silica gel through its 3' end. So the 3' end is attached to a silica gel, okay, silica beads. So that you can imagine where you have a column, and then on the column you have a lot of silica beads, and then you have your nucleotide attached to the beads, okay, so that even if you use water and then you wash the beads, your nucleotide will not come off, right? So it's physically attached to the beads. So that's why it's called solid phase. On here you have a protection group which protects the 5' phosphate, okay, well, in chemical synthesis, it's not exactly phosphate, as you will see. Okay? Now, first step is you're going to wash off this protection group such that you generate a hydroxyl group on top. Right? So obviously, this is already different from DNA, right? Because DNA, usually you have a phosphate there. Right? But here, what you actually generate is a free hydroxyl group. For DNA chemical synthesis, what you would add is actually this modified nucleotide. And let's take a look. This nucleotide has a base just like a normal DNA. So this is nitrogenous base, just like DNA, no problem. But what you notice is that the 5' end is protected, and then the 3' end is modified by this portion of functional group. What this will allow it to do is you can have a chemical reaction between the hydroxyl group and the hydroxyl group right here. What it's going to do is that your phosphate, okay, this phosphate looking thing, what that's going to do is it's going to react with your hydroxyl group on the silica beads. The rest of this molecule is gone. And then you create this DNA-looking phosphodiester bond. However, here you still have OCH3 left. And then what you would do is you would oxidize it so that you generate your phosphate-looking thing, but you still have the methyl group on top. And then what you would do is you would cut off the methyl group, and then essentially you generate your, this is N, and then this is N+1, okay? And then what you have is another cycle of adding a different base. The chemical synthesis of oligonucleotides is you start actually from the 3' end, and then you synthesize backwards. You start with the 3' end because the 3' is always attached to your silica beads, and then you're adding the next base onto here, and then you add another base, you add another base, and you add another base. Okay? So you're adding bases from 3' to 5'. Typically nowadays, primer synthesis, you can get pretty long, can get up about like maybe 100 base pairs or so. Okay. Obviously, why we cannot synthesize a whole gene using this method is because the efficiency. Okay, so every step you have a chemical efficiency. Okay, you have a reaction efficiency. So if you want to add a lot of bases, then efficiency gradually decreases, so it then becomes harder to generate. Typically, primers you can get, for PCR purposes, usually it's about 20 base pairs, 20 to 40, depends on how long you want your primers to be. So generally speaking, it's no problem in generating a little primer on a commercial scale. Okay, so currently each base costs about 30 cents US dollar, so roughly speaking, $9 in Taiwan, which makes biological research a lot cheaper than it used to be. So this is probably going to get cheaper and cheaper. Gradually, synthetic biology will become a very popular approach to solving different things. So this is the chemical synthesis route for making short oligonucleotides. Okay, obviously, you see how it's very different from biological synthesis of DNA. Okay, I think among the last things that I want to talk about is Sanger sequencing. Okay, Sanger sequencing is something that you probably have heard about back in general biology. In biochemistry, so here we focus on really what happens to the chemicals of the things that you're using in Sanger sequencing. Sanger sequencing was the first sequencing method that was very useful. Okay, prior to Sanger, we have other types of sequencing methods, but they were just not very powerful. But Sanger sequencing is still used today. Okay, so it's a good method. Now, Sanger sequencing is essentially done by adding this dideoxy nucleotide triphosphate to a PCR reaction, kind of. So what happens is that DDNTP, the dideoxy nucleotide triphosphate, if it gets incorporated to your growing chain, it actually permanently stops that chain from growing further. And the reason why it has two Ds in front of the NTP is that the 3' end, normally we have an OH group there, but in the DDNTP version of it, it's a hydrogen. It lost the 3' hydroxyl group, so that if the growing chain of DNA incorporates this guy in to it, then you no longer have a 3' hydroxyl, then you can no longer add another base onto this growing chain. Sanger sequencing initially was done by individually adding DDATP, TTP, CTP, and GTP into polymerization reaction using only one primer. Okay, so you don't have two primers. Note that PCR still contains the normal DNTP necessary for polymerization. Okay, so what happens is that you have 4 individual reactions. One would have the template. You still have the DNTP. You have the DNA polymerase. Okay, and of course, most importantly, you have a primer. In each of the individual reactions, you add a DDNTP. Okay, so in one reaction, you have DDATP. In another, you have DDGTP, and another, you have DDCTP, and so on. What happens is that the DDNTP will have a chance, and of course, everything is a chance. It would have a chance to substitute for the DNTP that it was supposed to go in that position. Okay? For example, let's say you have a sequence that is this. Normally, when you're adding, so let's say this is a 3' end, that's 5' end, and your primer is already bound. Okay? So normally, the nucleotide that's supposed to go here, go to this position, would be a DTTP, right? Normally, okay? Because normally you would have to take a DTTP, go here and make it a T so that you can continue to do your DNA polymerization. However, by chance, you will actually incorporate, if you have in the reaction a DDTTP, then what happens is that you will generate T, and then here's an H. You don't have the 3' hydroxyl anymore. So that the next CTP or dCTP cannot add on, okay, because you don't have the 3' hydroxyl to react with your next triphosphate coming in. As a result, you stop right here. In the 4 individual reactions, here, let's say you add DDATP, and every time when you're supposed to have a DATP adding to your chain, you may have a chance of DDATP. So as a result, you'll get premature termination. Okay? So what this band will tell you is that you added blah, 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 and then you added A. What this will tell you is you have blah, 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 and then A. Okay? So essentially, you do that to all 4 reactions, and then you'll get bands of different size, right? And if you have a good resolution, then what that will tell you is your sequence would be, here would be G, G, A, C, T, T, right? So then your sequence will look like G, G, A, C, T, T, blah, 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 blah. There you have it. That is the sequence for your particular segment of DNA. Okay? So this is Sanger sequencing. Okay? By using ddNTP, you can terminate the chain, and every time you terminate the chain, you're left with a particular segment of DNA. Okay? Now, you're probably wondering, wait a minute, but if you have ddATP in this case, if it terminates it here, then how come you still have this guy? Because remember, in the reaction, you still have the normal dNTP. By adding a little bit of ddNTP, you have a chance of terminating it. Does not mean that every single growing chain of your DNA polymerase DNA will be terminated at this particular position, because you'll have a competition between this normal dNTP and the ddNTP. This is essentially Sanger sequencing, right? Of course, the Sanger sequencing method has been improved, because previously you would have to do this on a gel, and you have to do 4 reactions to get a sequence of a particular DNA. Obviously, that's very annoying. So what people have done is by using a fluorescent tag on each of the different ddNTP. Okay, so essentially you will have a different color, okay, on each of the individual bases. What happens is, again, you still do your PCR reaction, which contains a template, primer, polymerase, the normal dNTP, and then colored ddNTP for each of the ddNTPs. Every time you, by chance, terminates the chain with either A, T, C, or G, then you have a small fragment. And then gradually you have larger fragments. And then you take this mixture, and then you run it using capillary electrophoresis. Okay, so we're using a capillary. Then you have a good separation, because each of the individual chain is different in size by 1 base pair. And essentially what you'll get is these things are coming out at different times, and then you have a laser that will be used to detect which color this thing is. Only the terminal base pair will have a color, right? Because all the other bases here are the normal dNTP, right? They're the normal ones. They don't have fluorescent colors. And when you shoot laser on it, it gets excited. It gives off a color. And then you have a detector that picks it up. So then you have very quickly a chromatogram that looks like that. Okay, so then you can immediately know the sequence of your particular segment. Okay, now the sequence that you get, of course, depends on your primer. Okay, so you are sequencing where the primer is. Okay, because ultimately, Sanger sequencing is still like a PCR reaction. Okay, however, you only have 1 primer. Okay, you don't have 2 primers. Okay, so you're not amplifying DNA. You only give it 1 primer so that you get something that looks like that. Okay, this is the primer. Every primer will give you a fragment of DNA. So currently, Sanger sequencing generates about 700 bases read per reaction. A lot of companies will tell you, no, we generate 1,000, okay? But in reality, you only get about 700 to 800 that are good readings, okay? Because towards the end of the reaction, you have a larger noise, okay? In those cases, it's very difficult to interpret which base is actually incorporated to your DNA. So roughly speaking, for every Sanger sequencing reaction that you do, you can get about 700 bases of good quality reads, okay? And this is used very routinely to sequence any plasmids that you make in the lab, okay? For example, this is what we commonly do in the lab. I have a vector, I cut the vector open, I put in a synthetic fragment of DNA in that I get from PCR, for example, and then I put it into my E. coli. My E. coli makes a lot of this DNA. I make a lot of this DNA. And then here is your oligonucleotide primer that you get from chemical synthesis. Okay? And this primer has a direction, 5' to 3'. And it's going to bind somewhere on my plasmid, give it to a company. And so I give the primer and the template to the company, and the company will run the Sanger sequencing for me, and as a result, I will get the sequence of the stuff that I put in, okay? Now, you're probably wondering, like, I thought you already know the sequence of this fragment, because otherwise, how do you PCR it out, right? That could be a legit question, okay? However, we always do Sanger sequencing to confirm the sequence is exact, because sometimes when we do PCR, you are likely to accumulate mutations. Okay? Sometimes you generate mutations during PCR, so this is why we have to always double check, confirm, and make sure that whatever plasmid that you're making is accurate. You don't have to know the exact chemistry of how the leaving group is going to help you attack the growing chain. That doesn't matter, okay? But what I do want you guys to know is at least that when you synthesize DNA using chemical synthesis, okay, it's actually done using a solid phase system where you have some kind of a silica bead. So this guy is solid, okay? And your first base is attached to it. Right? This guy is the first base. So let's look at this. Okay? So the first base is attached to the beads. And then when you add the second base, it's attached to the first base. Okay? And then you can wash it off. Okay? Wash off any reactive stuff. Like for example, after the reaction, you have to wash this guys out. Okay? After you wash it out, okay, your solvent, your base is ready to add another base pair, but you have to first remove the protection group, wash it out, you add this guy, and then you add the third base, you do it again, the fourth base, the fifth base, the sixth base, seventh base, eighth base. So you're adding the DNA from the 3' end to the 5' end. Okay? And then when it's done adding, let's say you want to make a primer that's 20 base pairs. When it's done, then what it's going to do is going to cut off, right? It's going to cut off this bond, and then you can elute this guy out, and then that is your primer. Okay. Now, when you chemically synthesize your short oligonucleotides, it's always single-stranded, okay? You don't make double-stranded primers. You only make single-stranded DNA, okay? I mean, that's how you are able to use this guy to bind to your template for PCR.